Hello, everyone. My name is Annabel. I'm from the University of California, and today I would like to talk about fitting Indel evolutionary model parameters to PFam protein alignments with this nice GPU accelerated framework. So to maybe begin by motivating air studies, it's probably obvious to everyone in the room that indels have a great functional impact on proteins. For example, this group in 2015 found that two proteins in the same organism with very similar structures and very similar sequences only differed in their activity site by one residue. And whenever they reverted that change from the protein on the left to match the single residue insertion on the right, they were actually able to recover the original, or probably not the original, but the similar protein function that was seen on the right. So clearly over long evolutionary timescales, these indel models are very significant. Maybe over shorter timescales, we also see that indels are equally as significant, as can be seen in the SARS-CoV-2 spike protein, which according to this study in 2021, managed to accumulate indels in quite simply a matter of months. And so knowing that these short indel events can dramatically change how air downstream protein functions, that kind of leads to questions like, how often should we expect these indel events to occur? How can we tell if our protein is rapidly occurring indel events? Probably more than we would expect. Luckily, we have statistical phylogenetic models, which offer these great tools and techniques such as joint estimation. We can look backwards in time by reconstructing ancestors, or we can move forward in time with the simulation of evolution. So we have all these great tools at hand, but with the caveat that they all sort of treat the short indel processes slightly differently. And that kind of motivates my overall question of what is the sort of the right baseline statistical model of how we should treat this short indel process, especially as we move to the era of AI and models with billions and billions and billions of parameters. What is sort of the kind of gold standard model-based solution that we can always refer back to and sanity check ourselves with? And so I will go briefly through the background of what kind of baseline models have been built for indels in the past. I will cover the experiments that we set up with the PFam alignments, and I will go through the different tasks where we benchmark different model-based indel methods. So to maybe take it way, way back to bioinformatics 101, these continuous time Markov chains are these simple, tractable, kind of gold standard models for modeling substitutions at any timescale. And in particular, this matrix exponential is the key to translating our instantaneous rate matrix, which happens over these small infinitesimal time points, to the actual finite time transition equations in this simple model of substitution. The TKF91, I don't know how to, anyways, the TKF91 model sort of extends this framework to start to include indels. And the foundation of this TKF91 model is that we're going to assume that there can be an instantaneous insertion or deletion, perhaps one residue at a time. The pros of this are that it's a very simple and tractable model. There are only two parameters, your birth rate and your death rate. And it also results in this pair HMM where the finite time transitions are exactly solvable. But of course, we know that biology doesn't very kindly introduce one residue indels at a time. And so in addition to this being mechanistically unrealistic, we sort of see the results of that this bias induces when we have these sort of gappy alignments and these inflated indel rate estimations. And so if the problem is that we're assuming single indel insertions, maybe we try to relax that assumption and say, okay, now we can have insertions of perhaps any specified mean indel length, and the natural maximum entropy distribution for specifying mean indel length would be the geometric distribution. This introduces two new parameters related to the lengths of those indels that are instantaneously inserted. But we sort of come across this problem of what are the finite time equations for these transitions? Because if we assume that multiple residues are now involved in the indel process, that kind of breaks our assumption that these sites these sites are acting independently, and it makes these transition equations really hard to solve. And so this sort of launched this whole quest to find the best geometric Indel process approximation. We have air class of models that introduce these latent variables, such as the TKF92 model, which introduces these indivisible fragments. And by introducing these latent variables, we're able to introduce these new independent units and find air finite time equations. But this does bake in a little bit more bias into our model. We could also have these class of approximations that try to avoid using these latent variables. In particular, we're very interested in this approach from Legend2024, where we match the moments of the true gap length distribution, which is, as we see in real data set, maybe not geometrically distributed, even if the instantaneous rate is. And so we try to match the moments of the true gap length to the best fit geometric Indel distribution. We are very excited to build off of this approach by extending this moment matching method with transducer multiplication. And to sort of briefly describe what's going on, we can describe a pair HMM that has air finite time variables indicated by f of t. We can multiply that by another pair HMM that has air infinitesimal time equations. That is the geometric parameters, and we can map the moments of this much more complicated pair HMM back to the three-state pair HMM that we all know and love by mapping the expected transition usages. And so in prior simulation work, we find that this H20 model is what we're calling it, produces parameter estimates that are pretty close to the original simulation values. In this graph over here on the right, we are plotting the relative entropy, and in this case, lower entropy is better because you're getting closer to the original simulated values. And our H20 model is that nice line in green. And you can also see that if we set the mean indel length to 1, which is to assume that we have these instantaneous insertions that are only of length 1, then the TKF91 indel model is exactly recovered, which is a great sanity check. And so by simulation, we see that the H20 model is the best approximation for the geometric Indel process. And that kind of begs the question of what if we move from simulation to real alignments? And so I'm also going to move out of DNA space. All of my examples were in DNA space. I'm going to move to protein space, and I'm going to benchmark these series of Indel models on PFam alignments. So I'm still working with this pair HMM framework. I am changing which models produce the transition equations, but I'm keeping the match position emissions and the insert position emissions constant through these tests. So I'm using the LGO8 amino acid exchangeabilities matrix, and for air and then the observed amino acid frequencies for guessing what comes out of the insert positions. I'm using PFam as a source for pair alignment trading data. We are interested in calculating the likelihood of pair alignments as opposed to moving to multiple sequence alignments just yet. So I started with about 20,000 multiple sequence alignments from PFam. And I also took the trees that were provided with the PFam dataset with the caveat that they were generated with this fast tree algorithm. We took a highly conservative pairing strategy approach from Frillo et al. 2022, where we start from the bottom of the tree and we select the closest family members together as pair alignments, and we sort of work our way up the tree to exclusively pair sequences together. We also assume, we bake in the assumption of reversibility by training on both pairs. That is, each sequence in these pair alignments get to take a turn at being the ancestor and the descendant, which leaves us with 1.2 million samples for model training and inference tasks. We're going to use the log likelihood function for the pair HMMs, which includes a term for the probability at emissions at match positions, the probability of transitions, and the probability of emissions at the insertion sites. And again, we are varying the part in the middle by changing what model generates our transition matrix. We also would like to not tie ourselves down with one specific divergence time, and we would instead like to marginalize that out. In order to approximate this marginalization, we use a discrete grid of divergence times Again, following the study from Frillo et al., we generate a time grid according to a geometric distribution. So given that we have models, that we'd like to benchmark, we have pair alignments from PFam that can serve as the training data, we're sort of off to the races. To begin with my first task, it is a standard machine learning task of which Indel model maximizes the likelihood of these pairwise alignment data set. We're going to divide our training set into a train test split, and we're going to see what happens whenever we train these Indel models for this training set and see how it affects the likelihood of the validation set. And so what we find is that the H20 model here, I've plotted the negative log likelihood, so lower is better. And I've also included on the bottom row as a reference for the lower bound, what happens if we just score based on match positions alone. This is akin to just tossing out every bit of all the indels we see and just scoring the based on matches alone with that LGO8 substitution matrix. And so what we find is that the H20 model produces the lowest log likelihoods but the KM03 and the TKF92 both do surprisingly well, and they get pretty close to what we have. If I look at the indel rates and the mean indel length that's produced by these indel models, we can actually see a surprising divergence. Even though the TKF92 model has a similar likelihood to these other more principled approaches, it vastly overestimates the indel rate, which you can see that the indel rate for TKF92 is much higher than TKF91, which we have already established overinflates indel rates already. Now that we know that the H20 model is one of the best fit approximations for a pairwise data set, now it's time to do another sanity check of are these indel rates actually biologically meaningful? And so to do this, I fit an H20 model on all of the proteins, both in the train and validation set, but I also took out the subsets of PFAMs that were labeled with a specific SCOP type, with SCOP being the database of protein folds. And we have these 4 different SCOP categories, and I am comparing the indel rates back to what is seen in the general protein data set. And so what I find is some sort of biologically intuitive results, such as that these type 1 globular proteins tend to have a lower indel rate and also a lower mean indel length, which makes sense because these globular proteins are under much more biophysical constraints to not change. On the opposite end of the spectrum, these non-globular or these unstructured proteins have a much higher indel rate, but also a slightly lower mean indel length insertion, which makes sense because they're not under the same sort of constraints and can freely and maybe more easily accumulate indels. And so given that our H20 model has the best likelihoods and the indel rates sort of seem to make biological sense, I wanted to look at a phylogenetic related task, maybe not yet building up the full phylogenetic tree, as that would require a multiple sequence alignment, but perhaps starting at the very bottom level with trying to find your phylogenetic match. And so I reverted back to the train test splits that I had before and trained on the exclusive closest pairs as I previously described, but I also, for the validation set, we now create all possible pairs within the PFam families. The idea is that we are going to train these indel models using that training set, and then we're going to evaluate the log likelihoods of those all possible pairs within the validation set, and use that normalized log likelihood score to predict which sibling that first layer of the tree should be paired with. We're going to declare it a true match if it's closest by the fast tree distance, with the caveat that fast tree, has its own bias baked into how it generates PFam trees. And so what we find is that all the indel models perform sort of similarly. The LGO5 and the RSO7 give us our greatest number of total matches, but we can see that if we look at the pairs that our models got wrong and ask how far away did you guess that original match, all of these models tend to perform very similarly. If they get a wrong guess, then they guess a similar distance away. And so kind of to wrap up, what did we learn from all of this? We have benchmarked these 6 different Indel models that approximate the geometric Indel process. Of course, this find your match test is potentially limited by this by referencing fast tree phylogenetic trees, because to begin with, the fast tree algorithm already tosses out these Indel positions. We find that the TKF92, the KM03, and the H20 models produce the best fit for our pairwise alignment data, but they have vastly different estimations of indel rate and length. I believe that the TKF92 model is probably a strong bias from the underlying assumption that there's these indivisible fragments, whereas the KM03 and the H20 yielded these more reasonable lower indel rates. And finally, for overall, in this whole task of fitting PFam pairwise alignment data, we find that the KM03 and the H20 are both really good candidates for a baseline indel model. We note that the KM03 by derivation is limited to assuming 1 to 2 indel events, while the H20 model has no restriction on the number of indel events allowed. Maybe the difference between these 2 models might be more apparent if we do pairwise DNA alignments, but I also suspect that we'll see differences as we move towards looking at full multiple sequence alignments. And so I would like to thank my advisor, Ian Holmes, and all the friends of the Holmes Lab. And as my highly scientific multiple sequence alignment suggests, I would love to chat and take questions.